Our group had the Krebs cycle. In order to have usable energy, eukaryotes use the citric acid cycle, better known as the Krebs cycle, to produce ATP. In eukaryotes, this takes place in the mitochondria, but in prokaryotes, it takes place in the cytoplasm instead. The cycle has the same starting and ending molecules and is a closed loop of 8 steps. Proteins, fats, and sugars can all be broken down into acetyl-CoA and enter the Krebs cycle.
The first step of the Krebs cycle. Acetyl-CoA bonds with oxaloacetate, a 4-carbon molecule that releases the CoA group, which forms a 6-carbon molecule known as citrate. Citrate is converted into isocitrate, isomer. This conversion is a 2-step process which involves the removal first, then the addition of water molecules.
In step 3, isocitrate is oxidized and a molecule of carbon dioxide is released. This leaves behind a 5-carbon molecule known as alpha-ketoglutarate. NAD+ is reduced to NADPH. The importance of the enzyme isocitrate dehydrogenase in the Krebs cycle is regulating the speed of the reaction. Step 4 is very similar to step 3, but in this step, the alpha-ketoglutarate is oxidized and the NAD+ is reduced to NADPH. A molecule of CO2 is released. The leftover 4-carbon molecule is used to pick up coenzyme A, which forms the compound succinyl-CoA, which is very unstable. The enzyme in step 4, alpha-ketoglutarate dehydrogenase, is crucial in the regulation of the Krebs cycle.
Coenzyme A of succinyl coenzyme A is replaced with a phosphate group. This is then transferred to ADP to make ATP. Not all cells use ADP to make ATP. Some cells use GDP to form GTP, which fills the roles of ADP and ATP. In this step, a 4-carbon molecule is produced. This is called succinate. In this step, succinate is oxidized. This forms another 4-carbon molecule called fumarate. During this step, an enzyme located in the mitochondria transfers 2 electrons to FAD, creating FADH2. This is done in the mitochondria so that the FADH2's electrons can be transferred to the electron transport chain.
Water is added to the cycle and turns the 3-carbon molecule fumarate into the 4-carbon molecule malate. In step 8, malate is oxidized to form the starting molecule of the cycle, oxaloacetate. NAD+ is reduced during this step as well into NADH.
In one turn of the Krebs cycle, 2 molecules of CO2 are released, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of ATP are produced.
And that's the
Krebs cycle!
3 minutes, maybe.